So in the book, we are in this section, is the chapter number 8, and we are going to use the same data set as yesterday. So ideally, if you were following the code, you should have the Visium brain object with some steps in the analysis, such as the filtering, the normalization, doing dimension reduction, then the nearest network, and then the clustering. Today we are going to use some of that information. So if you were not here yesterday, you also have this object available in the Zenodo files. So if you go to the section of downloading datasets here in the chapter number 2 under datasets and packages, you have the datasets to download, and we have provided a Zenodo link. So you can look for this for the session number, I think it's the day 1, session 5. If you download those files, you will find the Giotto object from yesterday. And if you download the day 2, session 1, which is this session, you can also find this Giotto object, but with additional steps in the analysis. Okay, so assuming that you have this object stored in the same path as we proposed yesterday, which is saving everything under results and then creating a specific subfolder for each session, we should have this object on their yesterday's session, which is the day 1, session 5. So let's go to RStudio. And first we need to load again the package. Let me make this bigger. So first we need to load the Giotto package, of course. Let's wait for everything to load. And then we need to use the function load Giotto. So yesterday for saving the object, we used save Giotto, which is going to create this folder. And then inside there is an RDS file. Now for reading again the object, we have load Giotto. Now, what's the difference? Maybe if you observe inside of this folder, you will see the RDS file, and probably you are wondering why we don't just use the read RDS, which is a very basic function in R. Well, the difference is that the function load Giotto will, yes, read the RDS file, but we'll also check that the expected structure of the Giotto object is correct. So in case that something was not saved in the proper way, or we are missing one of the slots for any particular reason, it will throw a message so that at least this object that you are loading, you will know that the object that you are loading doesn't have the proper structure. So that's why we recommend not reading directly with the RDS function, but using this function that we have created in Giotto. So that it will basically check that all the slots are there and that they have the right information. So once that we have read the object, we can just look inside. It contains the same information as yesterday. So since this is a processed object, it contains already both the raw counts, but also the normalized expression matrix and the scale matrix. In addition, we perform also the dimension reduction, so it contains the results from the PCA. Then, if you remember, we made an exercise just providing custom features. So we created a custom PCA, which is also here. Then we calculated the UMAP, the TSNE. We also calculated the nearest neighbor networks, and we calculated them using 2 methods. And it also contains the image, which is without any modifications. It's just the same image as at the beginning. So the first thing that we are going today is calculating genes that have a particular expression in each group. So for this step, you can use any clustering or any grouping information that you have under the metadata. So let's take a look again at the metadata of this object. So let's use the pdatadt function, and then we will provide the Giotto object. So this is how it looks like our cell metadata right now. So we have the basic information, which is the cell or spot ID, then whether these spots are in the tissue, then some statistics that we calculated. And at the end, we also added the Leiden clustering groups and the Louvain clusters. So if you have additional groups that you provided either manually or drawing probably different resolutions on the clustering, then you will find all of that information here in this metadata. So we are going to use basically these groups. It's important that you have categorical information, so you need to have groups here. And then we are going to calculate for each of these clusters, what are the genes with differential expression against or versus the rest of the clusters. Now we have two methods available in Giotto, Gini and Scran. So we usually perform both of the calculations to compare basically what genes we were able to find using one or the other method. And then usually you want to compare whether the genes that you obtain with one of the other method or probably either combining the results make some sense in the biological context. So let's first calculate using the Gini method. So for calculating the genes with a particular high expression per group, we have this function called find markers one versus all. So this means that basically it's comparing each cluster versus the rest of the clusters, and it will find the genes with the particular expression in each of them. So here we need to provide the Giotto object. Then we specify under the method which one you want to use, either Gini or Scran. And usually you would like to use the normalized expression for finding the genes. But of course, if you prefer to use a different one, you can select other of the expression matrices that you have in the Giotto object. Then you need to provide which column from the metadata you are going to use for creating the groups. In this case, we are going to use the Leiden clusters. And then you can select or customize what's the minimal number of genes that you expect to obtain per group. So let's calculate this Gini. I'm calling them as markers, but they are not always marker genes. A lot of times they correspond or they match with marker genes of cell types, but right now we don't even have the annotation of cell types. We only have clusters. So even if I put the name of markers, just let's keep in mind that not all the time they are marker genes. Okay, so let's take a look at the result. Basically, what we obtain from this analysis is a table with a lot of genes. The size of these a lot of times depends on how many genes you are asking per group, and basically they will be arranged in a ranking. So you will see. Let me see, where is this? Here. We have the rank at the end of the table. So probably just it's difficult to observe right now because I have the font very big. But basically, in the last column, you have the ranking for these genes, and then you can also observe what are the feature names. In this case, they are genes. But if you have proteins, you could also evaluate the levels of the proteins, and you will see here the IDs. So it's giving you the feature, then what cluster this is being detected in. Then it's giving you the expression, the normalized expression for these features in that particular group. So let's remember that this is at the level of the clusters right now. So this is not per cell, this is per cluster. And then basically other results after calculating the Gini method. So what matters the most to us is this ranking, basically. So we want to probably keep just the top genes, not all of them. Now, remember that here we established the minimal number of features that we were expecting per cluster, but we are not establishing a maximal number of features. So that's why we get a lot of genes. Probably you are wondering, oh, why I have 37,000 of results. Well, it's because we asked for a minimal number of features per cluster, but we don't have a top on the maximum. But you can select the top genes, the top results per cluster after getting this result, after getting this table. So from here, for example, you could play with different number of top N genes. In this case, I'm going to select just the top 2 genes per cluster, but you could select more. You could select the top 5, the top 10, and you will have more diversity on the genes. So I will select just the top 2 genes per cluster. So let's observe how this looks like. Basically, it's just a vector. In this case, I don't know anymore what cluster each of these genes corresponds to, but at least I know that these are the top 2 for, in general, the clusters. Now, how do we visualize the expression of these of these top genes? Well, we can create a violin plot. This is part of the Giotto visuals module, and here we are providing the Giotto object. Then we can provide the vector with the feature IDs. In this case, I'm specifying that I want the unique IDs from this vector, because sometimes it could happen that this top N genes between the different clusters have duplicates. So of course we don't need to plot the same gene twice. So that's why I'm using here the unique. So from this vector, I'm taking the unique IDs, and then I will also group the cells for the violin plot using the same cluster that I use for calculating the genes. And this is just basically for the text, where I want to put the text in the violin plot, and then some parameters for saving the plot. So let's run this violin. It's taking a little bit of time, but that's okay. It's not going to take a lot. Oh, okay. It's just that there is too much information, so in my screen it's difficult to observe. But probably if we take a look at the safe plot, let me see if I can find the safe plot. It should be here. So this is the result of our violin plot. So basically we have all the clusters. In this case, we have 19 leading clusters. And then it's showing each of the top N genes. Here we have on the right the gene IDs, and basically the distribution of the normalized expression for each of these genes. So from here we can start seeing some genes corresponding to particular clusters. For example, we can observe here that these 2 genes are basically corresponding to the cluster 19, because compared with the rest of the clusters, these 2 genes are showing the higher expression in this cluster. Then we can observe that apparently these 2 genes are coming from the cluster 18. This is from the 17, from the 16, and so on. Right? There are some of them that probably don't have the same level, since right now we are using the same scale in the y-axis. It's probably probable that some of these genes are kind of masked due to the scale. But of course, you can also take, once that you have the big picture of the behavior of these genes, then you could take individual IDs of your interest and plot them in a nicer way, probably in a smaller violin plot, even using the same function, but just subsetting some of the genes that you want to take a deeper look. But this is a nice way to at least have the big picture of what is happening with the top genes using this method and compare between the different clusters. Now, another way to visualize these results, again, we are using the same features, the same grouping, which is the latent clusters in the metadata, but we can create also a heat map. In this case, we have a color scale. Again, we are using the normalized expression here, but you can also select if you want to calculate C-scores and scale C-scores. And basically, each of those options allow you to visualize in different ways the same information. So the idea in general is the same, just comparing the expression of these top genes between the different clusters. So probably this is something that facilitates more the comparison, right? Usually the heat maps are very, very useful. So you have this plot as an option, plot metadata heat map. And then we can also use the scale expression so that we have a better comparison on what is overexpressed versus down expressed in your genes. But the difference between this plot and the previous ones is that we can observe in the spatial distribution how the expression of these genes looks like. So let's wait just a little bit. Minutes. Let me see. Oh, okay, there it is. Oops. Okay. Let me go to the save plot. Yeah, this looks better. So basically the difference, as I was saying before, is that in this case, we are observing, not the spatial distribution. We are using one of the dimension reduction plots for plotting what is the distribution of the expression. In this case, is the scale expression of each of these genes. So, for example, from I believe this is the UMAP, if I remember well. So basically, in the UMAP context, how is looking the expression on different spots or cells for each of these genes? So that can start giving us an idea of whether we observe the high expression in a particular cluster. So what you expect to see is that each of these genes is particularly expressed in certain regions of your dimension reduction plot. For example, here, this one seems to correspond with one of the clusters, while let's say this gene, for example, seems to be more higher expressed in this cluster over here, or this gene seems to be higher expressed in this cluster over here, right? Now, we can calculate, again, these results, but using the scran method. So the difference between the Gini and the scran method is that for the Gini method, not necessarily the genes are highly expressed in all the cells of the cluster. While usually the scran method gives us a better idea of the general behavior or the top genes in the cluster in general. So it's not just in a small group of cells in the cluster, but it's looking for genes that are particularly highly expressed in general in the cells in the cluster. So again, the result from this method is a table very similar to what we had before, but in this case we have different numbers. For example, we have the FDR, we have the log fold change, we have also the p-value, we have again the ranking, the feature IDs, the cluster where each of these genes is being highly expressed. And again, we are establishing in the arguments of the function the minimal number of features that we want per cluster, but we don't have a top. So that's why we are also getting around 30,000 of results. But using the ranking, we can organize them and just get the top end features of our interest. So we are going to do something similar to what we did with the gene method. We are going to get just the top 2 genes per cluster. So here we have just the vector of IDs, which is the top 2 genes. And then we are going to visualize using the same functions. So we are going to create again a violin plot, but this time we are going to use the top genes using the scran method. We are still grouping using the latent clusters. And this is just, these arguments are just for modifying the text here on the axis. Oh, let me open the file. Okay. So here we have the expression again of these genes. This is a different a different list of genes to what we observe in the Gini expression. But we also have, as you can observe here, a different scale. For some of them, it's more evident the difference. For example, this gene seems to be very highly expressed in the cluster 17. Let's see what's the behavioral no other ones. For example, this one is highly expressed in the number 6. And then let's see what else we have over here. Well, here we have a more diverse behavior. Oh, this one is more particularly from the cluster 2, and so on. Well, you can analyze these results with more time. But the basic idea is basically comparing again the expression levels of these genes between the different clusters. Then we are going to create again the heat map, this time using the scran genes. So we can observe the result like this. As you can see, and I think we were observing this in the violin plot, these genes are not always only expressing one of the clusters. They have a particular high expression, but that doesn't always mean that the rest of the clusters don't have some expression. So for example, I guess this gene was found as particularly expressed in the cluster 15, but as we can observe here, we also have some expression in other clusters. So that's why it's good to have the results of both methods, because you can compare after with the different plots and take the genes that you consider the better or that make more sense in the biological context for each of these clusters. Okay, and the last plot is creating the dimension reduction plot, but I'm showing in the scale the expression of these genes. Now, in this case, remember that we are using, for this particular plot, we are using the scale expression matrix. So that's why we have the scale with also blue colors, which is the under expression of some genes. So this is very nice, but because it allows us to compare what clusters have a high expression of some genes. For example, we have this one. It seems like these clusters over here have a high expression of this particular gene, while there are other ones that have even a very low expression of those of the gene. Sorry. There are other genes that probably are more dispersed, even if they are highly expressed in certain cells over here, with low levels over here. Here is another nice example where we have a high expression of this gene in this cluster, while we have a low expression in these other cells. In this case, this is another cluster with high expression, while these cells have a low expression. And you can take a look at the rest of the clusters here. Now, since this is a spatial data set that doesn't have a single cell resolution, we need to do an additional step, which is the deconvolution, for being able to annotate some cell types. Because remember that right now, even for calculating these Gini and Scran genes, we were using the latent clusters, but not cell types at this point. So if we want to create cell types in this spatial data set, we need to use as reference a single cell data set that provide us with the information in the patterns in the expression levels of annotated cell types. So we are going to download this data set, which is available using the Giotto data package. And for being able to download this particular data set, you need to use this name, which is the single cell RNA-seq of the mouse brain. So remember that for doing the deconvolution, you need to use the same tissue of the same tissue as in the sample for the spatial transcriptomics data set. So remember that we are using the Visium mouse brain. That means that we need to use the single cell of also the mouse brain. So I recommend that you download it and save it in the data folder under this day 2 and station 1. So I believe I already have this in my data. Let me just double check. If not, you can download it. It's not taking too much time to download. Yes, I have it here. So in this case, these files, even if they are zip files, you don't need to uncompress them because they are not folders. They are just 2 files. So R is able to read them directly. We still need to provide some instructions for creating a Giotto object, but the difference is that this Giotto object is not going to contain any special information. It's just going to contain expression information coming from the single cell RNA-seq and also the metadata, which will contain also the cell type annotation. So we need to specify where we want to save the results for this Giotto object. I suggest that you use the results folder and then create another subfolder with the day 2 station 1. You don't necessarily need to provide manually the Python path. Remember that if we set the Python path as null, then the Giotto functions will look automatically for the Python installation in your computer. But if you want to specify a very particular path in your computer, then you need to provide it either here or directly on the function for creating the Giotto instructions. So in the create Giotto instructions function, we are going to provide the directory for our results. Then we want to say whether we want to save the plot and show the plot or not. I will change this to true because I also want to show the plots right now. And then the Python path will be null in my case. Okay, so you can see that it automatically detected my Python path for the Giotto environment. And then I'm going to read or specify the path for my data. So I need to specify the path for the expression matrix, which is one of the files that I downloaded, and then for the metadata. So this is just redirecting to my folder here under data and then on my subfolder. And then I'm going to create the Giotto object. So I'm passing just the paths for the expression matrix and the metadata and just for the expression matrix. I will add the metadata later. So I'm passing just the path for the expression matrix and the instructions, which is saying that I want to save and plot my plots. Okay, so as you can see here, it created the Giotto object. It's also saying that it was not able to find any numeric or integer columns for the spatial location. And And then that for creating the cell metadata, it's going to use the first column that was able to identify as the cell ID. So let's see how it looks like, this object right now. So we have the spatial unit, which is at the level of the cell. Then the feature tag, I didn't specify a different one. So as default, we'll always create an RNA feature type. But when you are working with proteins, then you will need to specify that here when creating the Giotto object. Then in the expression level, it's only showing the raw expression matrix. And even if it's creating the slot of the spatial locations, I don't really have that information in this case. So it's just creating the basic framework for the Giotto object, but that doesn't mean that, that it's containing really the coordinates. In this case, it's not able to find coordinates for the single cell. Okay, so now I'm going to add the metadata. So if I ask now for the p data dt of this Giotto object, we see that we already have a lot of information about this data set. So this is part of basically the data that we downloaded. We haven't added anything else. So it's a lot of information. But the important thing for us is having the cell ID. In this case, remember that this is a single cell RNA-seq, so these are actually individual cells. And then something that we want to look for in this object is the cell type annotation. So in this case, that information is living in this column called class. Okay, so this is the column that we need for understanding what are the expression patterns in this single cell data, and then use those expression patterns for these cell types and guess what are the cell types that we have in the spatial data set. Okay, so now remember that in the expression of this Giotto object, we only had raw data, the raw expression matrix. So we need to still perform the normalization. So let's wait until this finish. So it's going to perform, normalized Giotto performs both the normalization and the scaling. So we are going to get the normalized and scale expression matrix at the end of this step. Okay. So let's just take a look again at the Giotto object. So here under the expression, we can observe that now we have the raw expression matrix, but also the normalized and the scale one. Okay. Now for calculating the markers from each cell type in this single cell RNA-seq, we have two options. The first one is just the ranking, using the ranking of the genes, basically the rank enrichment. And the other one is the DWLS, the convolution. So first, let's try the page or ranking method. So we are going to use the same function that we used before for finding the gene and the scran genes. But in this case, we are going to use the single cell RNA-seq object. And basically, we want to find what are the marker genes for each cell type. So we are going to use as the clustering group, or as the groups of my cells, the cell type annotation. And I'm going to use the scran method and the normalized expression. And in this case, I'm asking that I want at least 3 features per group or per cell type. So let's find the scran markers. Okay. I will get as a result the same table that I had before, the same structure, which is showing the ranking of each gene. Then I have the FDR, I have the log fold change, I have the feature IDs, and the cluster, which in this case is referring to the cell type. So now I can identify, for example, the gene with the highest ranking in the astrocytes, this gene. Again, we are going to select just the top genes for each cluster, or in this case, for each cell type. So I'm going to select now the top 10 genes, but of course you can modify this number and use the number that works best for you. In this case, I'm going to select the top 10 genes from these results. And then I'm just going to extract the cell types IDs from this table. Okay, so now I'm going to create a signature matrix. So basically what I want to find is, okay, I know that I have this list of marker genes per cell type. I need to understand now what is the behavior of these genes in the dataset, in the single-cell RNA-seq dataset. So it's going to create basically a matrix that contains this expression of the genes for the different cell types, not only for the cell type where those genes were found as marker genes, but it's also going to calculate the expression for the rest of the cell types. So if we take a look at this signature matrix, let's just print the first three rows and columns. So it looks like this. Basically, in the rows, we will have all the marker genes. So it's basically the group of the top 10 genes per cell type. So I'm going to have 10 genes per cell type. I think I have around 10 different cell types in this data set. And then it will calculate the expression of these. I think it's going to find whether this gene is expressed or not in each of these cell types. Yes, I believe that's what's happening. Okay, using these. Let me see. I think I already calculated this one. Yes, the signature matrix. So now I'm going to calculate the enrichment test. So we are going to use the run page enrichment using, now in this case, let's say, okay, let me clarify. So this signature matrix is describing the behavior of the genes in the single cell RNA-seq. And now using that information, I want to understand what's the behavior of those genes in the hypothetical cell types, but using my spatial data set, right? So basically, I want to use this knowledge from the single cell RNA-seq and apply it to predict something in my spatial data set. Okay, so this is going to take some time. Okay, there it is. Didn't take a look. Okay. So if we just print the Giotto object, now under spatial enrichment, we have a new result. This was empty before, but now we have information under the spatial enrichment, and that's the page method. Okay, so now how do we visualize the results? I want to extract the cell types available in my signature matrix. And then I'm going to use the same heat map function that I used in the past. But in this case, instead of showing the expression level per gene, what I want to do is observe how many cell types from the predicted behavior I have in my spatial dataset. So observe that I'm using, again, my spatial dataset. So what I want to describe here is not really the genes, even if here in the Y we have the text of genes. Here I'm not, you can modify the name of this Y-axis, so no worries about it. You can modify this name. But the idea here is that using the same function, I can evaluate the enrichment of each of these cell types in my clustering of the spatial dataset. So from here, I can start doing some kind of cell type annotation if I have a clear pattern. For example, I could guess that probably clusters 16 and 17 correspond to the ependymal cells, while, for example, the clusters 11 and 12 correspond to the oligodendrocytes, and so on. Yeah, basically the idea is that you can compare the enrichment of the predicted cell types using the behavior of the marker genes. Now, we can also evaluate the distribution of these predicted cell types in the spatial context. Let me see. Yes, we have a nice plot. So here, basically, I'm taking the position, the spatial coordinates of each spot from my spatial data set, and then I'm using the enrichment values for the different cell types, and this helps to also evaluate if this makes sense. If the if the high enrichment of the predicted cell types are corresponding to where anatomically you expect them to be. Basically, for this, you need more context of the biology in your sample, and of course, you need to know where in the past some of these very known cell types have been described. So this is basically these are the results of the page enrichment or rank enrichment method. Now let's use the spatial DWLS method. So here, the difference is that in the page rank method, we are predicting directly the identities of each spot, and we are basically saying how probable is that a certain spot corresponds to a particular cell type. But let's remember that this spatial data set doesn't have a single cell resolution. It's composed of multiple cells, around 6 to 7 cells per spot. So the DWLS method is going to estimate the proportions of the different cell types in each spot. So it's going to take into account that each spot is composed of different cells, and it's going to calculate basically how much of this spot correspond to each cell type. Now, of course, it could be that a spot is 99% of a particular cell type, let's say. So it's almost for sure that this spot is only composed of a single cell type. But when we have a region, specifically like in the transition of two different cell types in the tissue, then of course we cannot assume that the spot corresponds to only one cell type. It could be composed of 2 or 3 cell types at the same time. So that's why the DWLS method is very useful. So we need to, again, learn the behavior first from the single cell RNA-seq, and then we are going to use that knowledge of how the marker genes are behaving in the single cell RNA-seq to try to assign the proportions of the different cell types per spot. So we are going to create a signature matrix, and then we are going to use that signature matrix to run the DWLS, the convolution, on our spatial dataset. This step will take a couple of minutes to run, so don't worry if also in your computer it takes some time. I think probably I will stop this step, and I will load the object that already contains the results, just to don't let just to save some time. So I'm going to load randomization, load Giotto, and then results. Oh, I don't have it here. Wait a minute. Okay, so I believe this object contains the DWLS results. Yes, yeah. So you can keep running this step in your computer. It's not going to take forever. It's just that I'm trying to save some time for the session, but you can keep running this step. So at the end of running this step, what you should see when printing your Giotto object again is that after the page result, you also have the DWLS result under the spatial enrichment slots. So this result basically contains a table with the proportions of each cell type per spot. In fact, I think we can visualize that. Let me try to get the spatial enrichment from the Visium brain. And the name is DWLS by default, so I think we are going to get the right one. So basically the result of the DWLS, you can check that this is the spatial enrichment result here under the name. And you can also extract the results, for example, from the page enrichment. You just need to change here in the parameters the name argument. But here we have the result of the DWLS. So basically, this is a table with each cell ID. In this case, is our spots ID. And then it's showing the proportion of each cell type for each of these spots. So in this case, saying that, for example, in this spot, we have 0% of the astrocytes. We have a very, very low amount of neurons, but we have a high amount. It's basically just, it's pretty much the whole spot corresponding to peripheral glia. The same for this other spot. Then this spot seems to have pretty much the same cell type, peripheral glia, but it has a small proportion of these neurons. So probably in this case, these spots are coming from the inner section of a certain region of the tissue, and it's transitioning probably to the boundaries with that another cell type that could be happening. It seems that we have also some proportion of astrocytes here. So we could be on the boundary. This spot could be on the boundary between the peripheral glia with the astrocytes. And that's why we start observing some of the astrocytes. So this is a big table. It will contain on the rows all the spots and in the columns all the cell types. So how do we visualize these results? So basically, since we have proportions of cell types per spot, we can create a pie plot per spot and then position those spots in the spatial location, in their original spatial location. So it's taking some time to plot, but it's going to be there. Let me see if I have it already in the results. I'm sorry, this is not the session. No, I don't see it right now. Okay, let me go to the book. Here. So this is how the results should look like. Basically, we have all the spots from the spatial data set, and then each of these spots will be a pie plot. So as I was saying, it could be that in the regions where we are in the inner section of a layer of the tissue, we have basically 100% of these spot corresponding to a particular cell type. But then when we are in the boundaries with other regions of the tissue, then we have a main composition of a cell type, but also we start seeing certain levels of a different cell type. For example, here we have the transition between oligodendrocytes and astrocyte, which is in pink. And then this region, which is very, very well defined, it's particularly corresponding to neurons, but probably here we have a little bit of the astrocytes, so we could have a small layer here of astrocytes in the inner section. Then we observe here a region of very well-defined oligodendrocytes, but then in the transition or in the boundary with the epidermal region, we start seeing some small proportions of the oligodendrocytes, while the mainly composition is from the other cell type, right? So this is how the result from the DWLS looks like. Going back to the code. So this is basically the end of the DWLS method. Now, yesterday I was mentioning that when we calculated the Leiden clusters and the Louvain clusters, we were not taking into account the spatial locations of the spots. We were only clustering the spots, considering the expression levels of the genes. But we can find some expression patterns in our dataset. So before calculating the clusters again, we need to find those genes with a particular pattern in the spatial context. So we call them spatial variable genes. So in this case, we are going to calculate a network again, but this time instead of calculating a nearest network, we are going to calculate a spatial network. So we want to find basically those genes that have a strong correlation between their expression patterns with the location on the tissue. And we are going to use the KNN method. And basically, we need to provide our data set to run. Okay, so in this case, it's saying for me that the spatial network already exists because I loaded the object that contains all the steps of the analysis, but in your case, this message shouldn't appear. So if we look again our object, we will find this spatial network as part of the analysis. So here, before we didn't have, like in yesterday's Giotto object, we didn't have these spatial networks. We had nearest neighbors. So as you remember, we calculated, for example, the share nearest neighbors, we calculated the K nearest neighbors, but we didn't calculate the spatial networks. So now we have that slot here, and it's showing that we have available one result. If you calculate other spatial networks, for example, changing a little bit the parameters, you can assign a new name, and all the results will appear here in front of this first result. Okay, now let's visualize the spatial network. Where is this? It's not showing here. Okay, then go to the book. So this is how it looks like, the spatial network, basically showing the connections of each spot with their closest spots. So you can observe that, for example, here, there are some spots that are far enough to don't have an interaction. For example, here, these two spots are too far, so they are not going to show a spatial connection, basically, while this spot is going to have a spatial connection with their neighbors here, with the closest ones, but not with this one, which is too far. And at the same time, let's say this spot is not going to show a spatial connection with something over here because it's too far. So it's only going to have a connection with the closest spots, right? So that's the idea of calculating this network. So now the next step is calculating based on these connections. So basically the idea is that first we need to find the relationships between the closest spots, because that will help us to understand the spatial context of our tissue. Now we are going to calculate what genes are related to that spatial network context, and basically with that we will find which genes have more probability to present a spatial distribution, a particular spatial distribution with a high expression levels in that distribution. So if a gene is, for example, highly expressed everywhere, then that's not a spatial enriched gene, right? So we want to find the genes that have a high expression, but only in certain regions of our tissue. That's what we will call the spatial genes. Now, this step will take some time. So instead of instead of running this step right now, I will load the result. You also have that result in the Zenodo folder. So you can either run it. It's again not going to take forever, just that takes a few minutes, and I want to save time for the rest of the sessions. But it's perfectly doable in a local machine. No worries. It's not going to take a lot of memory. It's just that takes a little bit of time. Probably like around 5 minutes or so. So it's not forever. So in my case, I'm going to load this result from my files. So you will see, if you downloaded the Zenodo files, you will see this file called under the Day 2 Session 1. You will see the rank test .rds. In this case, we are going to read RDS. This is just a regular RDS, so we don't need to do the load Giotto. This is not a Giotto object. It's just a regular RDS file. Okay. So the rank test is again a table with the results of the analysis where we have the feature IDs, then we have the p-value, we have the adjusted p-value, and then we have the score of this gene and the high expression level. So let's take a look just to a few of these features. Right now, I'm not ranking them really, so I'm just taking them in the order that they appear here. Basically, I'm just taking the first 6 genes to see how they look like, how their expression looks like in the spatial distribution. So what you are going to get is this. For these 6 genes, the top 6 genes, I'm seeing that, for example, this gene is showing a high expression in this bottom region of the tissue. Then this other gene is showing a high expression in the out layer of the brain. This other gene is showing a high expression only in the center of the tissue. This gene is showing a high expression in this corner, like in the left of the tissue. This is also showing kind of an expression in the out layer, but more like towards the bottom instead of the top, and also a little bit on the center. And this other gene is more pretty much highly expressed in the center of the brain, right? So as you can observe, this makes sense. It's what I was saying before. You don't want to see a gene that has a high expression everywhere, but you want to see a gene that is highly expressed in a certain region of the tissue. And in fact, you usually see some overlapping with the clusters that you previously calculated. So we can start seeing some overlapping of this region with high expression with one of the layers that we observe in the latent clusters, for example. So this is telling us that we are on the right way. So now these are only a subset of genes. These are just the first 6 genes from our results. But now what we want to find is what genes from our spatial genes have a similar pattern, a similar expression pattern in our tissue, because probably they are related, probably they are involved in a certain biological function, or probably they belong to the same metabolic pathway. I don't know, probably they have a particular role in the development of this region of the brain. So those are questions that you can start asking when you find the genes that have a high expression in a particular region of your tissue. So what we want to do now is, okay, we found the spatial genes, the genes that have a particular spatial distribution with a high expression in those regions. We want to group the genes that have similar patterns. So we are going to calculate what is called co-expression modules. So first, we are going to subset from our long list of spatial genes, and we are going to take the top 500. Now, using these 500 genes, basically, this is just a vector. Let's take a look. This is just a vector with 500 genes with the highest probability of being spatial genes, basically. And now what I want to find is the correlation between those genes. And I want to group the genes with the highest correlation. So in this result, I have a table. I have a list. And let's go to the top. The first component of this list is the correlation table. So basically what is happening here is that for each of the 100 genes, it's comparing to the rest, to basically the whole 100 genes. So it's even comparing with the gene itself. So of course, if we observe the behavior of the gene versus the same gene, then the correlation, but in the spatial context, so basically it's saying, okay, where the first gene is and where the second gene is, and are they spatially correlated? Well, if you are comparing with the same gene, of course, the correlation is 1, right? But what happens if I compare this gene with another one? Then I start observing different numbers, right? So basically, I'm comparing what's the spatial location of these genes versus the location of this gene in the whole sample, and how much they overlap. And then it's also finding the expression correlation. So if I take the expression levels of this gene versus the expression levels of this gene, how much they match. Do they have the same expression levels, or are they completely different? Probably, even if they overlap in the spatial distribution, what if this gene is underregulated, and while this is overexpressed? So they are not going to belong to the same co-expression model, right? Because they have differences in the expression. So what we want to find is genes that have a good correlation both in the spatial and the expression context. So now we are going to identify, for example, for one of the features, this is just as an example, just to start seeing how our results look like. So we want to observe what are the features that have the best correlation to this gene. And we are going to visualize them using the spatial feature plot. And the result looks like this. So we have our gene as an example. And then we are basically plotting the 3 genes with the highest correlation, both in the spatial and expression context. So as you can observe, they match very well. You can see that the expression levels are pretty much the same. The location, of course, corresponds. For example, with this gene, with the second gene, we start observing a few differences. So we can see that even in general, it matches in this spatial distribution. We have some differences here in this inner section of the tissue, and not all the spots have the same levels, but they still have a good matching, right? And the same thing with this gene. So they have a good match in the spatial context, even if the expression levels is not exactly the same as the first gene, but they look very similar. Mm-hmm. They do look similar. Okay. So this is only one of the genes, just as an example. What we want to do is take the whole results, so the whole correlation table, and cluster the genes so that we are able to find all the genes that have similar behavior between them. And we are going to plot the results using a heat map. So the result of this heat map looks like this. Basically, what we are observing is in the rows and columns, we have the 500 spatial genes, which are the top spatial genes. And now what we are observing here is the correlation level. So, of course, we are interested on the groups of genes that have a highest correlation level because they are going to look like this. So if we were able to compare, for example, all of these genes, we will observe that they have a behavior similar to this one, let's say, or at least a similar behavior between each other. And then it's this function also is also showing cluster, because depending on their level of correlation, we can cluster those genes, and we can use different resolutions for these clusters. So right now, I decided to cluster them into 20 clusters, but probably I could reduce the number. And if I reduce the number, probably instead of having these 4 clusters here, I will be able to merge them into one big cluster that is basically representing this big square. Right now, it's basically separate them depending on small subclusters that we can observe like here. For example, this one in the corner, then we observe here kind of a square, then we have this other square. But you can play with a number of clusters just to find the best one. Then, for example, here is identifying another cluster. Here, identify like kind of a big square, but also 2 small subclusters. So that's why it's divided them in 2, and so on. The idea is that basically in this heat map, we observe the groups of genes with the highest correlation between each other. Now, we can also find the genes that are in these clusters, of course, because we want to see how is their expression and how is the expression distribution in our tissue. So what we are going to do is find the find and then plot the correlation and the number of the spatial genes for each cluster, because probably this is something that we are interested in. And once that we have grouped the genes, let me see. Let me show you a plot here that I'm not seeing right now in the RStudio, but the result of running this, The result of running this show spatial correlation features is that I'm grouping the genes with the highest correlation. So basically, instead of just plotting them as a heat map, as here, I'm basically now grouping them into the clusters. And then I'm showing the number of features that I have inside of each cluster and also their correlation level. So here we can see that, for example, in this cluster 16, we have probably around 25 genes. And then probably in this big group over here in the cluster number 4, I have close to 60. Probably I have around 50 genes corresponding to this cluster. So basically, this is the representation of the squares that we observe here in the heat map. Now that I have identified which genes belong to each of the clusters, I want to calculate the expression, the metagene for each of the clusters. So I want to find basically the expression for the genes that are composing, for the spatial genes that are composing each of the clusters. So I'm going to just run here. Here I'm basically getting the clustering, basically showing the spatial correlation features. Then I'm just extracting the genes, so the genes for each cluster, and then I'm getting the names for the genes. Now I'm going to create the meta genes for each cluster. Mhm. And now I'm going to plot the expression of the metagenes. So basically, I'm combining the expression of all the genes that belong to each cluster so that I can visualize them. Because if not, it makes very difficult. If I have the individual expression of each gene, then I will have to compare in this way, like using a spatial plot for each individual gene. And that's a little bit difficult, right? When I have a lot of genes. For example, imagine comparing all the genes from this square. So that's not really easy. So what I'm doing basically is combining the expression of all the genes corresponding to the same cluster. So basically the expression of all these genes, well, of all the genes from each of these 20 clusters. And then I'm going to plot the spatial distribution of those genes. So the result looks like this. Basically, what I want is to create just an individual spatial plot for each cluster and observe their expression. So let's say I have this cluster 13 and their expression correspond to this pattern, which is different to this pattern, for example. So what I want to compare is also whether it makes sense. The distribution of each spatial co-expression module, which is basically each of these clusters, and they should be different between each other. The idea is that we should observe differences. And it's actually what is happening, right? So if we compare the distribution of this one, it looks different to this one, different to this one, different to this one, right? But remember that these plots right now contains the combination of the expression of all the genes that had the same behavior, the same spatial distribution. Say. Okay, so now that I have these clusters, so I know that each of these clusters contain the information for spatial genes, right? So it's considering not only the expression levels of the gene, but also the spatial information, right? So I can basically recalculate the latent clustering and use that for, let's say, do again my annotation of the cells. So I basically can, let me see, I think I didn't run this code. This is just for getting the plot that I just showed you right now. And then I want to get the top genes here from my co-expression table. The co-expression table is basically showing this result over here. So it's just showing what genes correspond to each of the clusters that I found for each of the co-expression modules. And then I'm extracting the 13 most important genes in each cluster. So that's the idea. Since some of these clusters may have a lot of genes, then I don't want to keep all of them. I just want to keep the top genes from each cluster, because I expect that those are the genes with the highest, that will give me the highest information. So this is just a vector with the most important genes per cluster. And then using those this list of genes, do you remember that in the PCA, I can give a particular vector of genes to calculate the PCA? So now that I know that these genes have a particular pattern in the spatial context, I want to use them to calculate the PCA. And then I'm going to use them to calculate again the UMAP. I'm going to calculate again the nearest network. This time it's not the spatial network. This is like this, following the same steps that we performed yesterday, where we were just calculating the latent clustering. So remember that here we were calculating the nearest network. And then we use that nearest network to calculate the latent clustering. But this time, the latent clusters have information about the spatial context, because I'm not using just the genes with the highest expression. I'm not only using the highly variable genes. I'm using the genes that have a high expression, but in certain regions on the tissue. And the result of calculating the latent cluster is going to be stored also in the metadata. So in the same way as I had before, in the metadata, the latent cluster, let's say here, in the same way as I store the latent cluster in the Louvain cluster, I can now put the custom cluster. So the plot will look now like this. So it's again latent clusters, but now it's considering the spatial context. So it actually provides well-defined regions. This looks better than the latent cluster that we calculated yesterday, for example. Okay, I will try to hurry because I'm almost out of time. So just to show you, the UMAP, I can show you here. So basically, this is the UMAP now that I calculated using the spatial context, and I'm coloring the points also using the spatial context because I use the spatial genes. And they also are looking good. They look like very well separated clusters over here. Okay, so the last thing is that once that I have this information about the spatial context, I can also find spatial domains using the Haydn Markov random field model. So basically, we just run the modeling, calculating the HMRF. This is a step that also takes a few minutes to run. So I will load the result that I already have. And let me see. I think I have it again in my files for Zenodo. And I have the HMRF. Oh, sorry. Let me see. Oh no, it's actually just the path. Yeah, it's just the path. I believe it's just like that. Let me see if this works. No. Okay, I already have the results in the Visium brain, so no worries. So just going back here. In the, where I have it? I don't have the HMRF again. Let me just read again my Giotto object. No Giotto. Should have the HMRF. I believe it does. Let me see if I can visualize it. Yes, it's stored. It's just that I don't remember right now where I put it, where it's living in the Giotto object. But it's inside of the Giotto object for sure. Anyway, I will try to look what exactly is in this one, and I will let you know later. But anyway, the point is that, okay, just to summarize, this method is also trying to find these different patterns between the spatial expression and different regions on the tissue. So basically, it's going to correlate too. So instead of having all the calculation from having the calculation of the latent clusters, as we did a minute ago, what it's going to do is taking the spatial genes. So we actually need to calculate first the spatial genes, but then instead of doing a latent clustering, it's going to take into account those spatial genes, and it's going to look for their behavior on the tissue. And from there is going to create again the clustering considering the spatial context. So after you run this step, you can run it later in your computer. You basically have to store the result of getting the HMRF and then save it into the Giotto object. So basically here you are just saving the output, this previous output, put it into your Giotto object. And from there, what you are going to get is the cluster IDs, but coming from this method. Oh, I know where it was. It was on the metadata. So if we look at the metadata of our Visium brain object, we will have this column. Here is the information. So the result of running the HMRF method and then adding the results to the Giotto object is that we will get the cluster IDs in our metadata in the same way as we had the Leiden cluster and the Louvain cluster. So here we have different levels of the clustering. So we can observe multiple results of the cluster using this method, and then we can visualize them. So in this case, we are going to use the clusters corresponding to the HMRF with the number 40 here, and it's going to look like this. So as you can observe here, this is also a very well-defined clusters. It looks much better than what we calculated in the first session yesterday, just using the expression levels. So remember that the HMRF is also considering the spatial context. So it's taking the spatial genes and then learning and refining the clustering and trying to match the correlation between the expression levels of the genes and their spatial distribution. So as you can observe here, we have much defined clusters, right? Compared to even the Leiden cluster that we use with the spatial genes. You can observe that here still we have certain regions that are not completely well defined. For example, here we have an isolated spot coming from a different cell type. But then if we observe here, we have a very a better defined regions, right? And finally, I think I don't have too much time for going through this section, so I will explain it, but also you can play a little bit more with this. So we have integrated in Giotto an interactive tool that is basically launching a Shiny app. So it's running locally, so don't worry, you don't need to go to any server. This is running locally. The idea first is that you need to create a spatial plot, just a regular spatial plot, where you are providing these spots, and you will see them distributed in these in their spatial locations. Then, optionally, you can provide a cell color, but this is not needed. So if you want to just plot your original spots, that's also fine. But if you already have the information about the cell type annotation or the clustering using any of the methods that we saw before, you can basically create a spatial plot like this one, and then you pass that spatial plot to the Shiny app. So what is going to happen is that it will take this spatial plot and it will give you an interactive interface where you can plot regions of your interest. And the way to add new regions is basically just changing the name. You can also zoom in and zoom out. I can quickly just show you this. Oh. So we create this spatial plot. We have it here. So we have our spatial plot. Then we run the Shiny app. You can use this icon here in RStudio to show it in the whole window. From here, you can zoom into a specific region. So basically, you will crop or kind of subset your spatial plot if you want to select a very small region. So let's say I want to zoom in here to one of the corners. And then you can start plotting your polygon. Now, you can change the names here. So for example, region 1. So I'm going to start plotting the region 1 over here. And then you can zoom out again. So let's say that this was a small region. Now I can go back to the whole picture. And then I can select, for example, the region 2. I want to draw here my region of interest. And then if I want a region 3, I just need to change the name. And I will start plotting again. You can change the thickness of these lines and the colors in the arguments of the function that creates the Shiny app, that launches the Shiny app. So in case you want different, like thickest lines, for example, thickest polygons. So once that you have finished, you will click on Done. You will close this window and go back to your RStudio. And the output of this Shiny app is a table containing the X and Y coordinates for each point that you clicked on the app, and it's also grouping them by the region. So all the points that you clicked on the or for creating the first polygon or the first region, basically will have that label, region 1. Then all the points that you clicked for creating the second polygon will have the region 2, and the third one is the region 3, or the names that you assign to your different regions. Now, for storing this result into a variable that you will use in the future for performing downstream analysis, you basically just add the name of the variable that will contain your points, and you redirect the launching of the Shiny app. So basically here this is going to launch the Shiny app, but you are redirecting the output to a variable. So in this way, you can save your coordinates into a like the data frame instead of just seeing them in the console. Now, once that you have your data frame, we need to convert this data frame to the specific Giotto polygon object. So yesterday, George was telling you a little bit about the different objects that we have inside of the Giotto object, composing the slots. So we need to create a Giotto polygon object. Basically, we have a function called create Giotto polygons from data frame. So we are going to take our data frame with the coordinates. We need to call them as selections, because this is the default value that we identify the polygons with. But you can change this name. Just be careful when you are then manipulating or calling for the information, because this name has to be consistent when then performing the other steps. So right now I'm using the default. Once that I have created my Giotto polygon subject, I will add that information into the Giotto object here. Let me make this bigger, sorry. So you will add the information into the Giotto object. Then you need to integrate also the polygon IDs. So you are going to basically tell which cells in your Giotto object correspond to each polygon, and then you can extract that information again. So you can get the cells corresponding to each polygon. You will get basically a list of a table with the IDs of your cells or spots. You can either say that you want a specific polygon, or in our case was region 1, for example, region 2 or region 3. If you don't provide any name, then the cells from all the polygons that you selected will be provided. And then we also integrated some functions to make comparisons between the different regions that you selected. So for example, you can compare the expression of genes of interest between the regions. Here we have the mean score, the normalized mean score for comparing these expression levels. And then you could also, for example, find the kind of the marker genes, let's say, which is basically the genes with highly expression in each of these polygons compared to the other polygons, but also with the non-selected region. So it will compare with also with the background or the tissue. to try to identify which genes have a particular high expression. So here, for example, we identified a few genes. I basically took the top 2 genes with the higher expression per polygon, and I'm comparing their expression with the rest of the polygons, but also with the background. If you already have the cell type annotation or at least a cluster, for example, a Leiden clustering or the HMRF clustering, then you can also calculate the abundance of that cell type or that cluster per region. So in this case, it's just showing the percentage. So how much of the whole region is corresponding to this particular cell type? Then how much of the polygon 2 is corresponding to this cell type? How much of the region 3 is corresponding to cell type? And let me see what's the difference here. Oh, I'm just selecting a different column. So by default, it will look for the Leiden cluster column in your metadata. So if you don't provide any additional information here, it will look for the Leiden class column, but you can, of course, provide a different column referring to either the cell type of certain clustering. So in this case, I'm using the custom Leiden to show the difference. You can also isolate the regions that you have selected, and basically just to take a deeper look on what cells are composing those regions. You can also plot the expression levels on each of these regions to see if you have a particular distribution of high expression of certain genes of your interest. And finally, if you want to plot them again, just to remember how what regions you selected in the interactive app, you can plot again the polygons here using the plot polygons function. And that's pretty much it. So I suggest that if you are still running some code, at the end, you save the object because you are going to need it for tomorrow, I believe. So please save the object, or if not, you can also download it from Zenodo. But keep this object close to you because you are going to need it in the future. That will be it for me. Thank you so much.